Okay, thank you so much, guys. I think it's picking up the sound. So today we're doing lecture 3. We're going to do the ear, and we're going to do balance, and we're going to talk about the structures in the ear, the 3 different sections of the ear, the outer, the middle, and the inner ear. We'll talk about the structures in those locations and what they do, all right, and how sound is transmitted from the outer ear. This is the pinna. The outer ear is designed to gather sound waves and channel them to the inner ear through the middle ear. Okay, so pretty much the ear, of course, is for hearing and balance. All right, and there's 3. The external ear, which is called the pinna, and the grooves and the convolutions on the outer ear are designed to gather sound waves and channel them to the acoustic meters, external acoustic meters, which is the channel that leads to the tympanic membrane. All right. So all this design is not by accident; it's actually meant for that. So the external ear ends at the tympanic membrane, which is the eardrum. All right, and then you go to the middle ear, where you have the 3 auditory ossicles. There are 3 little bones that will aid the vibration of the tympanic membrane, channel the vibrations towards the inner ear. All right. So it's just an air-filled space in the temporal bone with auditory ossicles. Okay, and there will be the malleus, the incus, and stapes in that order. The malleus is, in touch with the tympanic membrane. Then the incus, then the stapes is in connection with the oval window. Okay, it's oval because it's like oval shaped on the inner ear. All right, and then it's going to move the perilymph in the inner ear. All right, on the vestibule side. Okay, and we'll talk about that. And then the inner ear. All right, it's filled with it's like you put a bag of a balloon. So the membrane will be the balloon, and you fill it with water, and you place it in a container or like a bucket, which will be the temporal bone, and then you put orange juice on the around the balloon. So that will be, the orange juice will be the perilymph on the outside, so that it cushions from the bone, and the water inside the balloon will be the endolymph. All right? And the endolymph is very high in potassium, and we'll talk about why that is. All right. So the inner ear, we start at the oval window, and the perilymph, all right, on the outside will be the first to be agitated, and then
outside of what?
Outside of the membrane, okay. Inside. So it will be the fluid that is lining the bone. Because the bone is, of course, rough, and it's not a surface where you can have a delicate membrane on it. So you have some fluid to lubricate. Just like the serous membranes in the body, the cavities, they've got serous fluid in between, so that when the heart is contracting and so forth, it will lubricate. So the same thing with the perilymph. It's on the outside. So this will be the bone, okay, the temporal bone, and it's a cavity inside the temporal bone. So what you have is fluid in here, then you put like a membrane-bound sac with fluid in it too, so that the membrane-bound sac is not beating against the bone.
What's the membrane-bound sac called?
That's the inner ear, and it contains the endolymph.
Oh, okay.
You see, the space within the temporal bone that houses the ear is divided into those 3 compartments. The first, the canal, external acoustic meters, which is the external ear. Then in the middle will be the middle ear, where there's auditory ossicles are located. All right. But when you get to the inner ear, the sound waves channeled by fluid. You see what I'm saying? So the fluid that is lining, it's like you put, just like I imagine you have a bucket, and that bucket is your bone. That's your temporal bone. And then you put orange juice in it. Then you take a balloon, okay, maybe one that is because it's kind of convoluted with all those things. And then you put water in the balloon, okay, and then you suspend the balloon in the bucket with orange juice. So the orange juice will be the parent leaf on the outside.
Yeah.
Okay, between the bone. So when the impulse comes into the oval window, it will hit the perilymph first, right? And then the perilymph will then hit the vestibular membrane. And we'll talk about a bit more about that when we get there. So that I don't want to introduce too many structures now yet. Just know that the ear in the inside is like a balloon with water inside, which will be the membrane, and it's surrounded by fluid on the outside to kind of lubricate it or to kind of cushion it from the bone. Yeah. Because you know when the membrane is vibrating against the sound waves and it's right against after the bone, it will probably tear. So that's why you have to prepare it. Okay, with the ear, you kind of have to track, you know, all these parts, and there's many parts. Each part is kind of divided into three, and three. The ear itself is divided into 3 sections. So you kind of have to keep track of it. All right. So we'll kind of go through it, and when we bring each section, you will then see how it's all working out. So the external and middle ear, of course, are involved in the hearing. And the inner ear is involved in both hearing and balance. See what I'm saying? Because the external and the middle ear, all they do is channel sound waves. That's it. And the sound waves are for hearing. But the inner ear is got the cochlea, which is involved in hearing, and it's also a membrane-bound, a section of the ear with liquid inside of it, surrounded by liquid on the outside. And then you have the oh, thank you so much, appreciate it. I was going to be looking for it. And then you have, of course, the vestibule, which is involved in balance, static balance, that is, when the body is not moving, and then the semicircular canals for dynamic balance. All right? So that's that and that's that. So we're going to divide up the ear. So, like I said, all these convolutions are meant to channel sound waves, not Southwest Airlines, sound waves, in there, and the sound waves will hit the eardrum, called the tympanic membrane, which will vibrate. Then it will hit the ossicles, starting with the malleus, which is this one right here, right? And then also the incus and the stapes. The stapes looks like a stirrup on a horse when you're riding, you know, the part where you put your feet in there, okay? And it vibrates against the oval window, right? Okay, this here, it's got nothing to do with hearing. It's a section of the facial nerve. What's the number for the facial nerve? The cranial, number 7, right? Okay, and it carries a taste from the anterior 2/3 of the tongue. Okay, it just happens to pass through there between the malleus and the incus. Okay, but it's got nothing to do with it. So it's called chordae tympani because it passes right in there. Right. So it's the facial nerve. Okay, it's just no room in the head, so it passes through there. All right, and then this is the cochlear part. You can see the cochlea is like a membrane, a labyrinth, pretty much with fluid inside of it, and then it's cushioned against the bone with the perilymph on the outside. Okay. All right. So, and then of course the vestibule. That's where you get the vestibular nerve here, and the cochlear nerve goes to the cochlea. The two will join to form the vestibular cochlear nerve, which is cranial nerve number. After that, 8. All right, there you go. So, and this is just an ear showing again a more realistic diagram. Okay, see the brain over there, the temporal bone. All right, and you can see how these things is all located inside the bone there. All right, so we're gonna go each of those parts. We're gonna talk about the functions. we're going to talk about how you generate the action potential and how it eventually ends up in the vestibular cochlear nerve to the brain, going to the auditory cortex, right? Okay, so the external ear is called the auricle, also called the pinna, like I said earlier on. Okay, it's the fleshy part. It's got elastic proteins in it, so you can stretch it a little bit. But the grooves inside of the external ear are designed to channel the sound waves to the inner ear, okay, to the middle ear in this case, and then to the inner ear. External acoustic meters is that channel that leads to the tympanic membrane, all right? And it is lined with hairs, and it's lined with ceruminous glands. Ceruminous glands produce earwax, and the little hairs kind of move the earwax towards the outside. All right? And what does the earwax do?
Keeps bacteria from.
Yeah, it attaches to particulate matter. It's not sticky. Have you ever cleaned your ears lately? And that's gross, you know, that thing is sticky. It's like really sticky. It traps any particles because the tympanic membrane is very delicate. You don't want to get bacteria and stuff like that over there. So you get that sticky wax, earwax, that will trap all those particles, and then those hairs kind of channel outside. It's almost like cilia, okay? And then, you know, once in a while you clean with the Q-tip. You shouldn't go too deep, because if you hit your eardrum, your tympanic membrane, that would be not so good. Okay, so ceruminous glands, right, produce earwax. There are sebaceous glands that have been modified to produce the wax. All right, and then of course the tympanic membrane, sometimes called eardrum, and the thin connective tissue membrane. Okay, it vibrates in response to sound, and then it vibrates against the malleus, which then carries the sound waves to the stapes, finally, which vibrates on the oval window. So this is the boundary between the external and the middle ear. Right? So we are good with the outside part and which parts are involved with the external ear and their functions, right? Okay. Now, you know, so the auricle, the temporal bone, and this just shows, highlights it, and you can look at it later on, and then acoustic meters and the tympanic membrane. Right. Again, you know, going further down, the cochlea, each of the areas is highlighted, and you can see vestibular cochlear nerve and the tympanic cavity, right, where the auditory ossicles are located in the middle, right? Okay. So, auditory structures of hearing. So, the middle ear. The middle ear, we said, has got those 3 auditory ossicles, right? So, to the outside will be the eardrum, and going medially towards the inside, then you have the auditory ossicles arranged malleus, incus, stapes, in that order. And you know what it spells? What it spells? Yes. So you can remember that order. If I was to ever ask about how the sound waves are channeled to the inner ear, you will need to know that order. All right. So, and then it will beat on the oval window. Okay. There's another round window that occurs on the scalar tympani. And I'll show you later on. So, these the malleus is attached to a muscle called the tensor tympani, all right? And the stapes is attached to a muscle called the stapedius. And these muscles, when you have extreme external sound, you have a lot of sound waves coming through the ear. So what these muscles do, they kind of pull on the malleus and the stapes to dampen the sound. Otherwise, if it's going to vibrate like crazy, you're going to mess up your fluid, you're going to blow up your ear. You see what I'm saying? Have you noticed when you go to a concert, you know, I like to stand in front. I only did it once though, I never did it again. I stand right in front of the speakers, where the speakers are really booming and it's loud. After that, I heard, you know, that sound, my ears were ringing the whole day. Okay, you know, until it tend to go off. Because what those muscles get used to do is that if you're listening to loud sound for a long time, they contract. And so when the external sound is gone, they kind of keep the tension a little bit, and then that's when you start to hear that in the absence of actual external sound for a while. So these are pretty good in dampening the sound so that you do not blow your ear into bits. Because again, those are delicate membranes with fluid in them. So these are very good. Tensor tympani attached to the malleus, stapedius, like the stapes attached to this, to the to the stapes. And then the auditory tube, which is a connector between the middle ear and the pharynx or the throat. Okay, so what happens is that, when you fly, you notice this a lot. If the cabin pressure is very high, okay, it's going to be pushing on your eardrum like that. And most of the time, you can cure that by yawning. Because when you take in air, it comes through your throat and it goes through the auditory tube, or eustachian tube sometimes called, and it equalizes that pressure on the tympanic membrane so that it's not being pushed in like that. Okay, so that's why sometimes they say chew gum, because when you're swallowing, as you swallow, the pressure of swallowing will put the air into the auditory tubes to equalize the pressure. Okay, so sometimes I ask about some of these parts and say, okay, what's the function of the auditory tube? Where is it? It's connected the middle ear to the pharynx or the throat, and it equalizes pressure on the tympanic membrane.
Also the Valsalva.
Mm-hmm. That's what it is. Okay, so equalizes pressure on the tympanic membrane. That's why it's also called pharyngeal tympanic or tube, or eustachian tube. Okay, so that's in the middle ear. We got ossicles, and then we got the the auditory tube or eustachian tube. All right. Again, these are the auditory ossicles, malleus, incus, stapes. All right. Okay. Those ones are real. That's an estimate of the size. If you take yourself a penny, and you can estimate the size of the auditory ossicles.
I have a plastic Angus.
Really?
Right here.
Yeah, it will do the job because, because as long as it will pass the sound waves, so they've shaped it like this and kind of put it in the things they can do these days. It's amazing. Yeah. Corneas, they can do that. They can transplant their lenses. So that's it. I just put that diagram so you can see. And this, of course, I'm sorry. I thought I was going to bring up. It's an otoscopic photograph. Like most of you nurses, when you go into the field, you probably be having to look through the ear. So you want to know what you're looking at when you look inside the eardrum. You see, this has got otitis media, which is inflammation of the tympanic membrane right there in the ear, and you can see the normal one. And this is, what is this? What do you think this is? What's that? What's that impression of? What's the first auditory ossicle? The malleus. Malleus means a hammer in Latin, so it looks like a hammer. Malleus. And this is the head of the malleus. See up here? Okay, so you see that if the membrane is nice and clear, you see this impression of the malleus in the background. So that's what that is. And of course, this is the wall of the external meters, right? And you can see the diseased ear with the infected. So that's why sometimes if you've got a cold, what happens? Your kid has got a cold, they get it? Ear infection. Why? I just told you the reason. That tube.
Eustachian tube.
Yeah, the eustachian tube connects to the throat, and the bugs for the flu, they're all over up in there, right? So that's how you get an infection, you get a flu and stuff, and you also get an ear infection. Yeah, it's all connected because of that. But it's important the auditory tube though, so that you can equalize the pressure on it. Again, just showing you so you can get an idea of what that otoscopic photograph is going to look like when you will be looking down the ear of some kid or whatever, or some adult. Okay, so very good. So this one just shows you those stapedius, you see, attached to the stapes, and then of course the tensor tympani attached to the malleus, and of course here is our facial nerve, the branch that controls taste from the anterior 2/3 of the tongue, right? So it's called the chordae tympani because it passes through the ear between the malleus and the incus. All right. Okay, so that's one of the things I wanted to point out there. Okay, so now we move on to the inner ear. All right. So the external ear is pretty much just the auricle and the external acoustic meters. The middle ear is pretty much the, and of course the tympanic membrane is the end of the external ear. And then the inner ear is that labyrinth, that the space in the temporal bone, and then the auditory ossicles, and that's it. The stapes is the last one that vibrates against the
That's
wing.
the middle area.
Yeah, the middle ear. The external ear with the auricle and the tympanic membrane and the external acoustic meters. The middle is the bony space in the middle, in the tympanic temporal bone, plus the auditory ossicles. And we know the function and the way they are arranged. Now the inner ear is one that's a little bit busy. It's also divided into 3 sections, right? The cochlea, the vestibule, and the semicircular canals. Each with a separate function. Okay? And each has a certain type of hair cell. Because when the sound waves are moving, or when your head is moving due to gravity and stuff, those hairs bend. And then that's how they generate an action potential that eventually goes to the vestibular cochlear nerve. Okay, so we'll talk about each part of the inner ear, the kind of hair cell you have in there, and how you generate the action potential. Okay, so again, it's like a bony labyrinth, just like the labyrinth over there. It's convoluted. So it's interconnected, and of course, fluid tunnels and chambers within the temporal bone. All right. So contains the vestibule, which I said is for static balance. Okay, and then the semicircular canal, which is dynamic balance when your body is moving. Okay. And then the cochlea is what that's involved in hearing. That's why we said at the beginning the internal ear, the internal ear is involved in hearing and balance. 2 types of balance. One when your body is moving and one when your body is not moving. And it's due to gravity, detecting the position of the head due to gravity. And also linear acceleration and deceleration. Okay, it's the vestibule. Okay, and then the cochlea for hearing. So there's a hair cell in each of these, okay, that generates the action potential. And then the membranous labyrinth, of course, like I said, the temporal bone being the bucket, and then you have a balloon inside filled with endolymph. Okay, and that's what the membranous labyrinth is, will be the balloon with the fluid inside. All right. So it's a series of membranous cells inside of that convoluted labyrinth in the temporal bone, filled with potassium-rich endolymph. Okay, and this potassium-rich endolymph is very important because when the hair cell is agitated, it opens potassium channels, and then potassium goes in. It's a very unusual situation. What normally creates an action potential? What is an action potential? What rushes into the cell? An action potential? Yeah, sodium. Sodium rushes into the cell. And what does potassium do? It rushes out. Okay? But still, potassium and sodium, they both have one positive charge. So the thing is, when you create an action potential, you just want the inside to be more positive and outside, and that's it. Whatever will do it, will do it. Even calcium can also cause an action potential if it goes into the cell because it carries positive charges. 2 positive for that matter. Okay, so in this case, this is the unusual situation when that happens. That's why the endolymph is rich with potassium, so that when the hair cells are agitated and they move, they open the gates, the potassium will rush in. And then when it rushes in, it will depolarize the membrane, and then you create an action potential, right? Which will cause the release of glutamate, which is a neurotransmitter. Okay, that's another, unusual thing because with the other things like muscles, it's acetylcholine, right? And then the acetylcholine will then generate the action potential in the vestibular complex. That's it. Right. So, space between the bony labyrinth, like I was saying, the bone, is rough, and then the membrane is filled with the pterygium on the outside to kind of lubricate that. All right. So it's the endolymph that is potassium rich, and that's important for generation of the action potential. Okay. So those are the parts of the inner ear. We got the dynamic equilibrium there with the semicircular canal. We got, in fact, the vestibule is kind of divided into the utricle and the saccule, okay, because they got two different kinds of hair cells, okay. The hair cells in the utricle are oriented horizontal to the base of the head, and the hair cells in the saccule are oriented vertical, perpendicular to the base of the head, like that, okay? Because this is detecting the position of the head due to gravity. So when you're like this, one of those hair cells is going to be agitated. When you're like that, you know what I'm saying? It's going to change shape. So you get two kinds of hair cells called maculae, and we'll talk about that, in a little bit. So, but I just wanted to point that because it shows the utricle there as part of the vestibule, okay? And the other part is the saccule, okay? It's just two different kinds of hair cells with the orientation of the hairs. And then, of course, the vestibular nerve. All right? Okay, very good. So, you can see how the bone, you see the bone here, and then you get the membranous labyrinth, all right? And filled with fluid inside. And then you've got the oval window here, where the stapes is vibrating against. And then you get the round window here. The round window, the purpose of the round window is that when the sound waves come through here like that, they go, and then they come out through here, and they are dissipated out through the round window. You know what I'm saying? Because if the round window is not there, when the fluid inside there is agitated, it's going to go like that and then bounce back and come back and disturb the sound waves again. You know what I'm saying? So you can't hear the sound. The initial sound you've heard is going to interfere with the incoming sound from the next waves. You see what I'm saying? Do you understand that part? If you didn't have the round window, then you cannot exhaust the sound waves. The sound waves will keep bringing back inside the cochlea like that. What's that?
Like echo?
Yeah, like an echo, if you will. Except an echo keeps like going because it's like when you're in a big room, it keeps projecting out. But with this, because the cochlea is a confined space, if you have sound waves coming in, they agitate the fluid, and you can imagine it goes and then it bangs against another wall, it's going to come back. The sound waves are going to come back again. And if somebody's still talking like I'm doing right now, the new incoming sound waves are going to interfere with that, and they'll cancel each other, you won't be able to hear. You see what I'm saying? So this is like an exhaust, just like the exhaust of the car. If the exhaust, the car didn't, it kept on, its fumes inside, as it's burning the fuel, it's going to become congested. You know, same thing. So this is like an exhaust ring. That's a good way actually. It lets the sound waves out so they don't interfere with the incoming waves. But, okay, so the cochlea, again, is got, it's kind of divided by like a Y, okay, like that. Like that, you know what I'm saying? So this will be the bony labyrinth, right? So this is the perilymph. The blue fluid is the perilymph. So that when the round window, the oval window vibrates, this is going to be hit first. And then this is the vestibular membrane, because this is the scalar vestibule. The top component, the cochlea, is divided into three parts. The one on the top is the scalar vestibule. Scalar vestibule. And the one in the middle, which is going to be covered by membrane, is going to be the cochlear duct. Okay? Where the hair cell for hearing is located, called the organ of Corti. Okay? Okay, so this is the organ of Corti, which is the kind of hair cell that's involved in hearing. Okay? And then this is the scalar tympani. Okay? Where the round window exhausts the sound waves. okay, and this is the basilar membrane, okay. So because this is the bony labyrinth, this is bone right here. It's the endosteum, which is the membrane of the bone. So this is going to be the perilymph. This is covered by membrane up here, covered by membrane down here, and it's the membranous labyrinth. So this is endolymph. The pink fluid is endolymph, okay? And it's the pink fluid that's bathing all these hair cells, all right? And I'll show you the orientation of the hair cells, because when the orientation of the hair cells, when they bend, they open up a gate. And you see that in a little bit. So scalar vestibular on the top and the vestibular membrane. So sound waves come through the perilymph, which was the order of the sound waves that was asked in the module that you guys had traveled with. So perilymph vibrates, then it vibrates the vestibular membrane. When the vestibular membrane vibrates, then what vibrates? Perilymph. And when the perilymph, the endolymph, sorry, the pink fluid, the endolymph inside the membrane, and the endolymph will vibrate, and then the hair cell will be agitated. Okay, when the base of the membrane is agitated. Okay. All right. So we'll come across that again, but I just wanted you to show you how the cochlea, which is the third part of the inner ear, is divided into three parts itself. The scalar vestibule on the top, the cochlear duct in the middle, housing the organ of Corti, which is the kind of hair cell that is involved in hearing, and then the scalar tympani on the bottom, okay, which opens through the oval window, right? So, okay. And then again, the semicircular canals also have those endolymph and perilymph on the outside. Okay, good. So, cochlea. We start with the cochlea for hearing. Then we'll go to the static and dynamic equilibrium. Okay, so again, like I said, these parts are all divided into 3, 3, 3, 3, 3. You understand? Okay, so the cochlea is spiral shaped. Why? Why do you think the cochlea is spiral shaped? Why not just a straight tube? You know what I'm saying? It's much easier. Yeah, well, it saves room when it's coiled up. And if it's coiled up like that, you can have a much longer tube. Otherwise, it will probably go through into your brain, and then it will start, taking some space. It's a small tube, but still, it means it's only contained in a small space and it's by itself. All right. So that's why it's coiled. So it's called the spiral-shaped canal. That's why sometimes the organ of Corti is called the spiral organ, because it's within the spiral cochlea. Okay. So it's spiral-shaped. So it's divided into 3 compartments. Again, everything is divided always into 3. Okay, so you just have to remember 3, 3, 3. Okay, so like I said, the scalar vestibule at the top, that's where the oval window vibrates, and this got the perilymph, and then, of course, the vestibular membrane separates the cochlear duct on the top, and the basilar membrane separates the cochlear duct on the bottom, which is in the middle, and then the bottom compartment will be the scalar tympani. Okay, scalar tympani. So the spiral organ, I normally call it the organ of Corti, okay? Organ of Corti. The guy with other sort of shape more like it. The organ of Corti is the spiral organ, is the same thing. You can use whichever you like, I don't really mind. Alright? So the organ of Corti has got the hair cells. So the organ of Corti has got the supporting cells, and then it's got the hair cell itself, and then of course the hairs on the top. Those hairs are called stereocilia. Okay. They look like microvilli. And I'll show you a nice picture again. So this is the hair cell, and the stereocilia are like that. Okay? They are not all the same length. And there's a significant, there's a reason why that is. It's very important when you open the gates, because there's a spring, an elastic protein that attaches the this kind of cilia, the stereocilia from the lower one to the top one, and then that one to the other one. So that when they bend like this, it's going to pull on the string, and then it's going to open the gate. See what I'm saying? When things are located like this, if I was to bend them, what happens? You see, there's like a distance. You see what I'm saying? This, and then it pulls. There's a spring between this stereocilium and this stereocilium. when they bend like that, then the distance is increased. Do you see? Like that. And then the gate opens. So that's how the gate for the potassium gate channels open up. You see how the stereo cells are arranged like that? So you've got the hair cell over here, then you got the supporting cells, okay? And then all that is the organ of Corti. And on top of the hair cell is a membrane called the tectorial membrane, all right? Over here on the top. Tectorial membrane, and the hairs touch that. So, and then this is endolymph inside. And I'll show you a picture showing you exactly how, this is an electron micrograph of the stereocilia, all right? Okay, and that's how they are arranged in the ear. You see what I'm saying? Again, when the hair is bent, that distance that goes, like this, it will pull on the string. There's an elastic protein here that is, denoted with a spring, because the spring stretches. So we see how when they bend, it starts to pull on the string, and that's how the gate potassium channels open up. You see, it's a mechanical, device. It's like a mechanical receptors, right? Just like photoreceptors are sensitive to light, these mechanical receptors are sensitive to mechanical force, right? So that's what opens the channel. And then because the endolymph is full of potassium ions, the potassium ions will rush into the cell and depolarize the membrane. Okay? So that's how you generate the action potential. Because of the orientation, you see how over there it's blown up, so you can actually see how when the hairs bend, they open up the gate. Do you understand how the action potential is generated in the ear? With the hair cells? If I ask it in the exam, you can answer that question. I say, explain how the hair cells in the ear generated action potential. You'll be able to do that, right? Yeah. And it makes sense too. It's kind of like more intuitive than it is cerebral, Kind of makes sense. So we're going to look at this and how the sound is, the quality of the sound comes about in terms of the pitch, in terms of the loudness and so forth. We'll talk about that.
Sound waves strike the tympanic membrane and cause it to vibrate. Vibration of the tympanic membrane causes the three bones of the middle ear to vibrate. The footplate of the stapes vibrates in the oval window. Vibration of the footplate causes the perilymph in the scala vestibuli to vibrate, which in turn causes displacement of the basilar membrane. Short wavelengths from high-pitched sounds cause displacement of the basilar membrane near the oval window. This movement is detected by hair cells of the spiral organ, which are not visible in the animation. Long wavelengths from low-pitched sounds cause displacement of the basilar membrane far from the oval window. Again, this movement is detected by hair cells of the spiral organ, which are not visible in the animation. When the vibrations reach the perilymph in the scala tympani, they travel to the round window where they are dampened.
Okay, so the first thing that was introduced is the pitch of the sound. Okay, let's say you're hearing a lady. Most ladies have a high-pitched sound, so it's going to hit close to the round window. So these are the high-pitched sound waves, okay, which are close together, because pitch is frequency of the sound waves. And then the low-pitched sounds, like if you're hearing heavy bass or whatever, it will hit the tympanic, the membrane much later in the cochlear membrane, much later in the almost towards this. This opening here is called the helicotrema, the opening that unites the scala vestibuli to the scala tympani. It's called the helicotrema. Okay, so that's the pitch. Pitch is frequency, and then we'll talk about volume also, which is the amplitude of the sound waves. Volume. All right. Okay, very good. All right. So auditory function. So pitch is a result of the frequency of the sound waves. Okay, the frequency of the sound waves is how because they're coming together. Okay, like that is a low, high pitch sound. The frequency is high. That's low pitched sound with the same volume. And I'll show you a graph later on. So high-pitched sound is going to create more action potentials, right? Because it's coming high, and it hits the basal membrane much earlier. Okay, low-pitched sound, it takes its time. By the time it gets to the end of the cochlea, you know, so the frequency of this, you know what I'm saying? By the time it, you know, the low-pitched sound waves, the reason why they hit the basilar membrane much later in the cochlea is because they're coming one and then, so it will go and then come back. But these ones are coming quickly, so they quickly hit the, you know what I'm saying? It's high-pitched sound. You see the frequency and how the thing separates, how you make. Because in the exam, I'm going to ask you to explain how you determine the quality of the sound and how you get high-pitched sound, how you get high volume as well. Okay? All right. So volume is the amplitude or the size of the sound wave. All right. So again, now I'm going to do that's high pitch, high volume. See what I'm saying? High pitch, low pitch, high volume. It is higher than this. So the amplitude of the wave is from here to here. So that's the amplitude. Okay? So here the amplitude is only this. But there's a third quality to the sound called timbre. Right? It's like the overtones of the sound, the quality of the sound. Remember when we talked about the bones, we talked about the air spaces inside the nasal bones, like the ethmoid and sphenoid, and facial bones, and so forth. We talked about you know, the maxilla having those air-filled spaces. And what did we say those air-filled spaces do, apart from making the bone light? Do you remember what I said they do? Those spaces. They act as sound resonant chambers, right? So that the sound echoes inside those spaces and gives the quality of the sound. So the timbre, which is kind of like the resonant qualities or the overtones of the voice, is you know, a result of those sinuses, okay? And how the voice kind of echoes inside of your sinuses. That's why when you've got a cold, what happens? When you've got a sinus infection, your voice changes, right? And that's the quality that we're talking about, where the sound kind of resonates inside those air-filled spaces. Okay, so timbre is the resonant quality or the overtones or just sort of the richness of the voice. All right. So I need you to be able to explain all this and how we determine pitch and how we determine you know, volume, and then the timbre, of course. All right. Got to be able to explain that. So this really shows you what I just drew earlier on. This is a sound wave, low pitch and high volume. Okay. And this here, high pitch, low volume. High pitch, high volume. Because you see, here's the amplitude, the size of the wave determines the volume. And then, of course, low pitch, low volume. You know, high pitch, low volume, same volume as that. And this is high pitch, low pitch and high volume. Okay? So that's how you determine the pitch and the volume. Pitch is frequency and volume is amplitude of the sound. Okay, so now we do the hearing, some ways are funneled through. Now we're kind of starting from the beginning and going through now the structures. The external ear, okay, gathers the sound waves, goes through the external acoustic meters, and then it will vibrate against the tympanic membrane, and then the Himalayas, the Indus, and the States in the middle ear. All right. The stapes will vibrate against the oval window. Okay. And it will agitate the fluid in the scalar vestibule of the cochlea. And then when the scalar vestibule is agitated, it will then agitate the vestibular membrane, and then also the basilar membrane, because it is agitated the perileaf, right? Once the basilar membrane is agitated, because the perileaf is agitated in the scalar vestibule, also the basilar membrane will agitate, right? And then when the basilar membrane agitates, which is where the hair cell is located, the organ of Corti, then the hair cells will bend. And then when they bend, because of that elastic protein that connects the stereocilia, it's going to pull and open the potassium channels. And then, because the endolymph is full of potassium, it's going to rush in to the cell, and then it will depolarize, and then it's connected to the cochlear nerve, the cochlear ganglion, where that nerve will synapse at the cochlear ganglion, which is right next to the hair cells, and then it goes into it will produce glutamate, which will agitate, or which will then stimulate the action potential in the neuron that reaches the brain, right? So external ear up to the tympanic membrane, right, and then oval window, right. So these are the things to note, and then the auditory ossicles, and then the perileaf in the scalar vestibule. This is the scalar vestibule perileaf, right? The scalar vestibule is separated from the cochlear duct by the vestibular membrane, so that's why the vestibular membrane will vibrate first. Then when the vestibular membrane is vibrating, because it's vibrating against the endolymph inside, then the endolymph will be agitated, which then agitates the basilar membrane. The basilar membrane is where the organ of Corti is located, and then you see what I'm saying? So that's the deal that you had trouble with in the module in terms of mapping the sound waves all the way to the inner ear. Okay? So you see how the vestibular membrane comes in, then the endolymph will do that. When the endolymph in the cochlear duct is agitated, then the basilar membrane is going to be agitated. Then finally, it's going to be dissipated through the scalar tympani on the bottom, through the round window. See what I'm saying? So that's where you're getting there, and then auditory ossicles in the MIS, sequence, and then 4 oval window, and then the perileaf in the scalar vestibule of the cochlea, right? And then 6 is the vestibular membrane, because the vestibular membrane demarcates the perileaf from the cochlear duct. Then the perileaf, the endolymph inside the cochlear duct will go, okay? Once the endolymph in the cochlear duct goes, then the basilar membrane vibrates, okay? And then the basilar 7, 8, all right? And then the organ of Corti goes, all right? Which is the spiral organ, right? And then the organ of Corti goes, and then the hair cell generates an action potential. Okay, done. Number 10. Right? Action potential, not anatomy and physiology. Action potential. Can you map this and get 10 points in the exam? Anyway, okay. Maybe you map 5 of it, but external ear coming to the tympanic membrane, auditory ossicles, right? The steps vibrates against the oval window, then the scalar vestibular perileaf will be agitated, then the vestibular membrane, because scalar vestibule. Vestibular membrane will then be agitated, which then agitates the endolymph in the cochlear duct, which then, because it's connected to the basilar membrane on the bottom, then it starts to vibrate basilar membrane will then agitate the hair cells because the organ of course is slapped right on top of the basilar membrane. When that happens, then you know the hair cells will bend, and then you have an action potential. Get to open. That's it. Okay, so that's how it's mapped, the sound waves. So what happens is that you know the nerve that's connected to the hair cell, will synapse with another neuron at the cochlear ganglion.
So it's a possible application.
Well, I could ask you to map that, and get 10 points if I ask you to do that. But this time I'm going to reduce the points of the exam from 100 to like 50 or 60 points, and you have less long answer questions. So I'm going to expect a little bit more from you. Like you will be answering 3 long questions only instead of like the 6 or the 8 that we used to do. Yeah, I know that. I want you to kind of sort of sometimes attach reasons as to why things happen, not just tell me what it is that's happening, but tell me why it's happening also. You get points for it.
So more than just like 5 points. Bullets anyways.
Yeah, we're still going to be bullet points, but at least be able to kind of synthesize some reasons for some of these things. Okay, I'm going to ask you to do a little bit more critical thinking about it, not just to be able to tell me back the facts, but to tell me, okay, you put this together, okay, why that is, and why is it that it happens like that, that kind of thing, And we'll talk about those things in class too. Okay, it's not like I'm just going to expect you to do it and we don't talk about it. Okay, so in those instances when those situations arise, and I'll tell you about it and say, okay, this is when I may ask you to give reasons, and I'll give you those reasons. All right. So we are continuing on with our conduction of our action potential, right? Okay. So the hairs are bending, the stereocilia are bending, and then again, because of their orientation, one is shorter than the other, it will put some tension on that elastic protein, and then it opens up the potassium channels. Okay, and like I said, it's an unusual situation because normally sodium is one that depolarizes the membrane, but all we need is a positive charge. It doesn't have to be sodium. There's nothing magical about it. It's got the same positive charge as the potassium has. So potassium will work too. Okay, so that's what happens, and then the calcium, potassium ions will enter the hair cell as a result of opening the gate, and it will depolarize, and the depolarization will release glutamate. That's when the glutamate will take the message to the second neuron at the cochlear ganglion. Okay? Because the cell bodies are located there. And then generates an action potential in the sensory neurons associated with the hair cell. Alright? So it's a little bit much, but okay. And then, of course, the round window will dissipate the sound waves so that we do not hinder the incoming waves. But at least you understand how the gates open up and how you generate the action potential, right? And then the neurotransmitter in this case is glutamate. Glutamate is a neurotransmitter. It's an amino acid, and it's also a neurotransmitter. Right? Just like acetylcholine. Okay? That's the neurotransmitter that is used in this situation. Alright? Okay. So this is kind of showing you how, again, it's kind of repetitive. When the sound waves come in, they agitate the tympanic membrane, and then agitates the malleus, the incus, and the stapes. So number 2 will be the auditory ossicles in that order. Right? The last auditory ossicle, the stapes, vibrates against the oval window, which agitates the fluid in the scalar vestibuli. Okay, scalar vestibuli, right? Okay, and then that, depending on the pitch of the sound, it will agitate the basilar membrane via agitation of the vestibular membrane, the endolymph, and then the basilar membrane will be agitated. And then when the basilar membrane agitates, then it agitates the organ of Corti located in the cochlear duct. Right? And then when the organ of Corti is agitated, it then bends the hairs, and then that's when the gates open, and then the sound wave continues on to the scalar tympani and is dissipated through the round window. Alright? And that's it. Okay? Alright, so this shows the frequency. So the frequencies of the sound waves it starts off high at the beginning. So this is high pitched sound. If you notice, like on a speaker, whenever I buy loudspeakers, I look at what the frequency response is. So if it's got very high frequency, so you get your trebles, you get your little cymbals and so forth, and if it's very low, then you get your bass. It can go up to 50 or so hertz. That's like very low. Okay, so 200 hertz is low pitched sound, is the bass sound that hits it over there later on. Okay, so this is 7,000 hertz to about 200 hertz. It's kind of like and this is the corresponding frequency. See how it is, and then it's kind of increasing. You see, so high pitch to low pitch, high pitch to low pitch sound. All right, frequency, high frequency, high pitch, low frequency, low pitch. All right, so it's kind of like an inverted tube. If you start off very high and then it goes low towards the end. So the neuronal pathways. So the vestibulocochlear nerve first goes to the medulla, right? It goes to the medulla oblongata, and then remember we talked about it. We talked about the corpora quadrigemina, and we said it's got 4 bodies. 2 of them are the inferior colliculi, and we said they're for hearing. Okay, and then 2 of them are the superior colliculi for sight. Remember that? So once the impulse gets to the medulla, it goes to the inferior colliculi. Okay, it goes to the inferior colliculi. Okay. Remember we said all the senses, they go to the thalamus except what? Smell. It's the only sense that doesn't go there. Everything else goes to the thalamus. So if I was to ask for any sense and ask you to talk about the neuronal pathway and you don't mention the thalamus, if it's not smell, then it's wrong. It has to go through the thalamus. It has to. So medulla, then it goes to the inferior colliculus, then it goes to the thalamus. Then the thalamus directs where it goes to the auditory cortex in this case, right? So the thalamic neurons will take the cortex, because the thalamus is like a triage, it will determine what sensory information it is, and it will determine which part of the brain will best deal with it. So in this case, it's the auditory cortex that will best deal with it, kind of somewhere in the temporal lobe of the brain, right? Okay, so that's where the impulse will go. So again, if I ask you to map, and then of course, you need to control the sound waves. The efferent neurons will go to the cranial nerve and be responsible for controlling those muscles that emit sound, which will be the stapedius and the tensor tympani. See what I'm saying? To kind of control them. All right. It's kind of like, when the sound stops, and the sound has been perceived, and then you can tell them to stop. Because when something is vibrating, it will kind of keep on kind of vibrating, and you have lingering vibrations. So once you have perceived the sound, you have converted the information, you send the impulse back to stop them. See what I'm saying? Otherwise, they will stop much later on. And you see what I'm saying? You understand that? So you've got to send that message to the to the tensor tympani. So so that you know those muscles, and the stapedius, and it will tell you where it's located, to stapes, and then of course the malleus. You see what I'm saying? So medulla, inferior colliculi, thalamus, and then auditory cortex, and then and then you send a message to stop. So we go to the vestibule now that deals with static balance. So it judges the position of the head due to gravity when the body is not moving. Also, it will detect linear and deceleration and acceleration. That's the vestibule. So that's
Is this the part that deals with like motion sickness?
yeah, and motion sickness together with the, because motion sickness is kind of like, you know, when your body is kind of moving, but it's like you're not really moving, and the vessel you're located in is moving. I get motion sickness really bad. So it's detected by your inner ear, and it causes problems because your body is not actually moving, and it, you know, because it will be sitting right there in the same position, but then your position is changing because you're in something that's changing position. So that kind of messes people up, like me. I get motion sick real bad, and I can't even contain myself. It's like everything is, it's the worst feeling ever. I didn't know about it, and when I went on a boat that was doing this, my God, I suffered. Okay, so those are these are the ones that detect that. Yeah.
Dramamine is awesome.
Yeah, it kind of knocks you out.
It makes you sleepy though.
Yeah, that's what I don't like about it. I want to experience my thing here properly, my cruise or whatever it is. So anyway, so the vestibule, again, you know, these things are always divided into three things. You know, like I said, the utricle is got those, you know, horizontal hairs to the base of the head, and then the circle has got vertical hairs to the base of the head. And the kind of hair cells that is located in the utricle and the circle is called macula. That's all. The hair cell that, you know, just like the organ of court is the hair cell for hearing in the cochlear duct. That's it. So the maculae, that's the hair cell. Okay? That is found in there. Now, remember we said the tectorial membrane bounces on the top of the organ of Corti. The hair cells in here is the otolithic membrane. It's like a gel with little calcium stones called otoliths to give weight, because again it's gravity, right? So it puts weight on those hair cells, so that when you move like this, then that weight will move with the stones, and when you move like this, and then that's how it agitates the cells. Okay? That's all. So the otolithic membrane helps with that. It's just a gel that gives weight on the head, so that when the weight moves like this, it will move the hairs. Because the hairs are too thin, too fine to move, you know what I'm saying? Due to gravity. That's all. Okay? So it gives the mass to it, so it moves in response to gravity. When the hairs bend, again you get the situation, and then you open the gates, and then you generate an action potential. Okay? So these are the three parts that I want you to know that are involved in the vestibule. Okay, the kind of hair cell in the vestibule is called maculae. Okay, and there's two types of them: one with vertical hairs and one with horizontal hairs.
I think that was spelled wrong in the Learn Smart module. It actually said like macula.
Macula is singular.
Oh, okay.
Yeah, macula is
I
singular.
thought that was in the eye.
Because this is Latin, you know. Without the e, it's singular. Macula is singular, and maculae plural for both the utricle and the circle. Okay, so it's macula is correct, but it's just means one, singular. All right. So, and then the hair cells, the otolithic membrane will give the weight, and I'll show you a picture of it. You see, the hair cells are embedded in this, and you see the same situation. Okay, except the longest hair cell for the maculae is called the caudocilium. The longest stereocilia, you see, these are stereocilia, and the longest one is that it's called a caudocilium. So it's just got a fancy name because it's the maculae. Okay, it's the hair cell. The longest one is called the caudocilium. Okay, so that's in the vestibule, and you got the circle and the utricle, which have got horizontal and vertical hairs respectively. Okay, and this is what it does. You see, when your head is like this or when your head is like that, it will move the hairs, and then you generate an action potential. So dynamic balance. So we know the parts of the vestibule. Okay, we know the parts of the cochlea. All of these parts are divided 3, 3, 3, 3. Okay, I don't know why, but that's how we did it. Okay, so this one evaluates movements of the head. Okay, if you notice, when those guys are dancing and they spin around, You ever thought why they don't get dizzy when they go like this and they do these things and they spin like very fast? Yeah, because when we were young, we used to go like this and go dizzy and then we fall down. But those guys don't get dizzy because, because we're talking of the same circular canals and there's fluid in there that moves and agitates the hairs. So when they're going to spin in this direction, they flick the head to the opposite direction so that the fluid starts to go there. So when they start spinning in the opposite direction, the fluid comes back and it detects zero motion. So they don't get this. Okay, that's how they do it. Because otherwise they would really get so dizzy, it's not even funny. We just fall off, the dance is finished. Okay. So semicircular canals, those are the ones so-called because they're semicircular and they are around in the three planes, the X, Y, Z plane. Okay. So because they detect motion in all the directions. Okay. Because the X, Y, Z plane is kind of like that. In X, Y, Z plane, that's what they call 3D. Okay. Okay, so you get it, you see the 3D. So that's how there's a semicircular canal here, semicircular canal here, semicircular canal on the three planes. So that's why there's three of them. So there's the semicircular canal widens over here to form a housing called the ampulla. Okay, and the ampulla houses the hair cell that you find in the semicircular canal called the crista ampullaris. Okay? So we got the organ of Corti for hearing and the cochlear duct. And then we got the macula for static balance in the vestibule. Okay? Then we got the crista ampullaris in the semicircular canals, which is located in the ampulla for the animal balance. So those are the three hair cells. We are done. Okay? So this is the hair cell that you find in those bumps. So there's another bump here, bump there, there's a bump here, bump there. And that bump is ampulla, and it houses the crista ampullaris. And we've talked about something that always lies on top of those hair cells. We talked about the otolithic membrane for the maculae. We talked about the tectorial membrane for the organ of Corti. Now we talk about this, the cupula, which is a gelatinous mass on top of the crista ampullaris hairs. That's it. Okay. And so when the head moves, the end of the in, the semicircular canal moves, then it agitates these hairs, and then that's how you detect movement of the head. Okay. So everything is divided into 3, 3, 3, 3, 3. Even the ear itself, middle, external, and inner. 3, 3, all the time. Okay. And just know each hair cell that occurs, and the parts of that hair cell, what lies on top of it, and so forth, and you'll be fine. And you just have to learn 3 of them. That's it. That's it. Okay. So there is the ampulla with the crista ampullaris in there, and the cupula, which is the gelatinous mass that goes onto the hair. And you see the same arrangement of the stereocilia. They're all like that because again, when the hair bend, the same generation of axial potential. Same thing. Okay. So when you have to limit once and you're done. So this guy is doing that. But I don't know what a gymnast. And that's how the hair cells move. So the neuronal pathways for balance again, going through the vestibular part. So the macula for static balance and the crista ampullaris for dynamic balance will be involved here. They generate the action potential. Again, it goes to the medulla. Okay, that's where it's processed. Then it goes to the spinal cord because balance involves, some spinal reflexes going to the muscles and back and forth. So it's going to involve the spinal cord. The cerebellum, we talked about how the cerebellum is one that teaches you fine movements. Okay, and learning to dance and all these things. It's balance. All that is located. So it goes to the cerebellum before it goes to the cortex. Okay, for balance, which is probably in the same temporal lobe. Okay, and then the nuclei that control the extrinsic eye muscles. Have you ever tried balancing with your eyes closed? It's kind of hard, isn't it? So the sense of balance of the ear also involves the eyes. So it's going to control the eye muscles too. Okay, so if we're talking neuronal pathways for balance, you want to bring in the spinal cord. Okay, you want to bring in the cerebellum. Okay, and you want to bring in the eye muscles. Okay? Because balance involves all those things. Okay? And that's it. All right. And then, of course, proprioception depends on the visual input. That's why you need the extrinsic eye muscle. All right? And that's it. Thank you, guys. I appreciate it. I can give you 5 in the lab if you like. Okay? And we start at 5 past. Okay? Thank you so much. I appreciate it. We just had a few problems with the mic, but that's already done. Hopefully, the recording is good.